Hello dear students. I am sure you must have understood all the concepts of genetics till now. Till now we have studied about Mendel's monohybrid cross, and based on the two laws, that is law of dominance and law of segregation, and we studied the exceptions based on law of dominance. Then we studied dihybrid cross, and on the basis of dihybrid cross we studied the law of independent assortment. We have also studied how the Mendel's work remained unrecognized, and later on how different rediscoverer studied his work and uh, explained his concept. We also studied the chromosomal theory of inheritance. Today, let us take up some exceptions for the law of independent assortment. Because when there are more than one pair of genes, the interaction becomes more complex. So, we will be dealing with these three exceptions. First is polygenic inheritance, epistasis, and linkage. So, today I will be dealing with basically polygenic inheritance and little bit about epistasis, because epistasis is not there in detail in your syllabus, but polygenic inheritance is there, and linkage is in great detail, which we will take up in next lecture, because it's a big topic, big explanation. Okay, so let's start with first concept, that is polygenic inheritance. So the first concept, that is polygenic inheritance. Polygenic inheritance is also called as quantitative inheritance. Mendel basically dealt with only qualitative inheritance. What is qualitative inheritance? Qualitative inheritance means when the character or the phenotype is controlled by one pair of gene and the phenotype is same whether the genotype is homozygous or it is heterozygous. For example, if you take stem height is tall because of the dominant gene capital T. So, if the genotype is capital T, then also the plant will be tall. And if the genotype is capital T small t, in that case also the plant will also be tall. So there will be no difference phenotypically. That is called quantitative inheritance. But later on it was found out that the number of dominant genes also play a different role. That is, more the number of dominant genes, more prominent will be that phenotype. This means if more dominant genes are there, that phenotype will be more stronger. And if less number of dominant genes are there, the phenotype will be not that stronger. It will not be that prominent. That is called as quantitative inheritance. So here we will be dealing with quantitative inheritance or polygenic inheritance. In polygenic inheritance, it is not that there is only one pair of gene which is controlling one character. There may be more than one pair of genes present at different locations, that is, they are non-allelic, and they are controlling the same character. So that is called as polygenic inheritance. Here in case of the skin color may be capital A, capital A, capital B, capital B, capital C, capital C. Means three pairs of genes are controlling one character, that is the production of melanin in the skin. So this will be a black color. Likewise, when all the three are in recessive, that is small a, small b, small c, small c, it will be white. So here the color will be dark. In this case, the color will be white. And when there is a cross, the F1 will have capital A small a, capital B small b, capital C small c, it will be intermediate color. And when there is a cross between this type of genotype, then a large number of progeny will be produced, types. 64 combinations may be there, and those 64 combination will result this type of phenotypic ratio. There will be 7 types of phenotypes, which will be there in the ratio of 1:6:15:20:15:6:1. That is white, very light, light, intermediate, dark, very dark, black. And the genotypic ratio will be so confusing. You can see here 1:2:1:2:4:2:1 and so on. So it is called as polygenic inheritance. Let us see this polygenic inheritance with the help of animation first. So now let us see again these polygenic traits through this animation. Well, this polygenic traits, as you all know, they are the traits which are coded for by 2 or more genes on different chromosomes. That is, they are non-allelic genes, which means there are more than one pair of genes which are coding for one trait only. For example, you can see here the skin color is controlled by 3 pairs of genes, that is chromosome number 3, chromosome number 5 and chromosome number 8 together. So more the number of dominant genes, more darker will be the color. Less the number of dominant genes or more recessive genes means the skin color will be light. Therefore, it is called the continuous variations. So you can see there may be 7 types of combinations, and we already studied the combinations may range from complete white to complete dark in the ratio of 1:6:15:20:15:6:1. Apart from the skin color, there are many other traits which show the polygenic trait, polygenic inheritance, which is called as quantitative inheritance. Let's find out which are all those traits. The second trait in human beings is eye color. In eye color also, there are different types of eye colors depending upon the genotypes of mother and father. So there are 2 pairs of genes which are controlling the eye color. That is capital A, capital B, and small a, small b, small b. So because of this gradation, there will be difference in the eye color. Likewise, the same goes for the hair color also. The hair color also may range from very dark, that is black color, to complete light color, and in between there are so many different shades of hair color. So all these traits, they are polygenic or quantitative traits which are in a continuous variation. After polygenic inheritance, let us now study a little bit about another concept called as epistasis. Epistasis is a phenomena when the expression of one pair of alleles is influenced by the presence of some other non-allelic pair of genes. So in that case, there are 2 types of genes. First are called epistatic genes, which are influencing, and second are the hypostatic genes, which are being influenced. For example, in case of Lathyrus odoratus, that is sweet pea, the expression of the flower color can be influenced by the presence of another epistatic gene. So instead of 9:3:3:1 ratio, they get the ratio of 9:7. So that is called as complementary gene ratio. Similarly, there is supplementary genes. Supplementary genes are those genes which, when not present, their expression will be different. But when they are present, their expression changes. So, the ratio is 9 is to 3 is to 4 in place of 9 is to 3 is to 3 is to 1. So, that is seen in the comb type in hen. Okay, so these are some exceptions. So with this, we have completed two exceptions, that is polygenic inheritance and epistasis. In the next lecture, I will take up the process of linkage and recombination.